Now let's welcome Jakub for the next presentation. Jakub is a biomedical engineer and currently a PhD student in laboratory of X-ray, micro, and nano computed tomography at Brno University of Technology. His area of research is image processing and quantitative analysis of micro CT scan. He has the capability to contribute to new findings in the field of medicine by providing deeper sight into samples under investigation, thanks to the higher resolution provided by the micro CT scanners. Today, he will present a novel approach how to utilize image processing method to evaluate spinal fusion process. The title of his paper is Evaluation of newly formed bone after lumbar integral diffusion surgery using X-ray microcomputed tomography. Welcome.
Yep. Thank you very much for a kind introduction. On the beginning, I will maybe a bit disappoint you because since we are in the machine learning and deep learning section, I will not talk about machine learning and deep learning today, but I think I can give you a good assumption on how to utilize, say, standard image processing techniques and methods in order to evaluate the quality of newly formed bone after the lumbar interbody fusion surgery. So at the beginning, what is it, lumbar interbody fusion? It's a type of surgical procedure which is done in order to stop motion or prevent motion between two or more vertebrae and connect them as one fused unit. There are several reasons to undergo this surgery. Mainly, it's to relieve pain and pressure in the spine due to some degenerative diseases or to treat spinal stability in case of some scoliosis, etc. The procedure or surgery is done in the following manner. Firstly, the intervertebral disc is removed and is replaced by some kind of medium provides the fusion, and after several weeks of the fusion process, two or more vertebrae are connected into one whole unit. Regarding the materials for the fusion, there are several types of them. Usually, as a reference, are used bone grafts, which is a small piece of bone harvested from the patient itself, usually from iliac crest. This is called autograft, or it can be allograft. This is the same piece of the bone, but harvested from some donor or cadaver. Or there are actually researches of novel biomaterials which can provide this kind of fusion instead of the bone grafts. There are several demands on those materials, mainly osteogenic, osteoinductive, osteoconductive functions of those biomaterials. And here in these figures, you can see how does it look, the 3D model of vertebra after the removal of the intervertebral disc and after the placement of this kind of biomaterial before the start of the fusion process. And regarding our data, we obtained 15 samples of porcine fused vertebrae fused by 3 different materials. And the task was to select or evaluate which of those 3 materials has the best ability or potential to provide the vertebral fusion. Regarding the materials, 5 samples utilize the bone graft. Then there is a novel bioimplant based on a ceramic scaffold enriched with fibroblast growth factor 2. And in the third group was the same kind of material used, but the fibroblast growth factor was replaced by polyphosphate bioimplant due to the high cost of the fibroblast growth factor. And here you can see micro CT scans of those porcine fused vertebrae, one representative sample from each group. Thanks to the utilization of micro CT, we were able to achieve the voxel size of 40 micrometers. It means that much more detailed scan compared to the clinical CT scanner. And now the task was to evaluate the quality of the fusion. So there are available multiple imaging techniques like histology or plain radiography. We went in the way of microcomputed tomography. And according to the state-of-the-art methods, we found out that most of the methods are based on some visual assessment by the radiologist or by an expert who can utilize some grading system, like giving a grade from 1 to 4, for instance, to grade the quality of diffusion based on the visual assessment. So, for instance, here you can see grade number 1, where is clearly visible the fusion process, or here grade 4, where no fusion occurred. And because we wanted to do this comparative study of biomaterials, we decided, or we wanted to develop a novel algorithm which works objectively, it means automatically, and gives as a result just one accurate number on how good the fusion quality actually is, not those categories. So there is a proposed approach how can be this achieved. Firstly, the CT data are registered according to top cranialis, bottom caudalis orientation of the fused vertebra. Then the sample is binarized using OTSU thresholding methods, and are applied some morphological operations in order to fill the holes in vertebra, which are done by pedicle screws and plates, which are supporting the vertebra during the fusion process. Since this sample creates one connected unit, is detected the area of lumbar interbody fusion, the diffused area, by fitting of ellipses, one ellipse for each vertebra, and according to the cross section or intersection of the ellipses, its major axis is estimated the region of interest. The dataset is then cropped according to this region of interest, and utilizing advanced iterative watershed transform are those two vertebrae separated to find actual surface of each vertebra and joint area. You can see here in this figure that since those two surfaces are extracted, consequently are constrained to just one voxel wide area surface, and based on this we can estimate, we call it tiger value, which is a abbreviation for trabecular ingrowth ratio, and it's basically just the ratio between the surfaces of vertebra and the joint area. You can see in red here means how successfully those two vertebrae fused, divided by the location, how successfully the vertebra could fuse, but actually did not. And this gives just actually one number representing the ratio of the fusion. Consequently, we wanted to compare such kind of results with the state-of-the-art methods, the visual methods. Created this kind of graph where here is the tiger value and there are the grades according to two selected state-of-the-art methods. The blue one is according to Tan et al. I will show you the references later. And you can see that different samples can be categorized by different grade by the two different approaches. So that's the thing we wanted to unify, to find just one number which can represent the fusion. And in general, you can see that the tiger value correlates exponentially with the state-of-the-art methods. We also found out that there are some outliers, like here, where the tiger value is close to 0, but there is grade 2, which means that there actually is some development of novel bone. But we found out when we looked closer to the data that the trabeculization occurred only or was attached only to one of those vertebrae. So the vertebrae could still move and the fusion process was not completely done. And we wanted to go a bit further and explain the whole process of the fusion. So we did say morphological analysis of the newly developed bone. That way that we created a manual reference of the fused area based on 6 completely different samples. According to those segmented masks, we did like one average model, and this average model we fitted into individual samples. And after this kind of registration, they're able to describe the newly developed bone according to just classical parameters describing the bone morphology. We found out that there are, in some cases, a correlation between those morphological descriptors and the Tiger value. And when we looked further for those correlations and morphology, we found out that the fusion is created in that manner that the biomaterial or the bone graft starts to expand as one connected unit and then connects two vertebrae and create the one single unit. Also, was possible to compare the three groups of materials where were used different mediums for ensuring the fusion. We found out that the categories have like pretty similar values. Maybe just the trabecular number value is higher in group B, which means that the fibroblast growth factor, which is in group B used, has the ability to increase the number of trabeculi and just expands the fused area more rapidly. Or in like as an additional value could be evaluated some kind of those morphological parameters to, for instance, compare here the trabecular thickness between the area of fascias intervertebralis, the vertebral body, and the area of the newly developed bone. So in the end, to make some conclusion, here is presented an approach how can be visualize and assess the fusion quality of fused vertebra as a one single number, as an objective approach. This was demonstrated utilizing a micro-CT technique on the ex vivo scans of pigs' fused vertebrae. And according to this approach can be also, let's say, made a comparison between different groups of materials used for this kind of vertebral fusion. So, like another thing, we found out that samples in group B, where was the fibroblast growth factor used for the fusion, proved better results than the reference bone graft. And regarding the future work, we would like to expand this methodology also for clinical scanners and clinical CT data for real patients. And for this, because the spatial resolution of clinical scanners is limited compared to the micro CT where was conducted this study, we would like to quantify more parameters to describe the bone even further, like bone mineral density, or detect some fractures and abnormalities in the bones. So that's all, and I'm looking forward to answer your questions you might have. Thank you for that.
Thank you very much for the interesting presentation. Do you have any questions to Doctor? Yes, please.
You use the bicultural algorithm to search exercise with
I can maybe show here. Yeah, it's like this kind of image is a region of interest, and we use the watershed to separate in this part where it's used, or where the bone actually fused. So the result of watershed are, let's say, two volume or subvolumes of the interest, upper and bottom vertebra, and in between emerged like one voxel wide area where actually is the bone fusion. Not pretty easily, but it can do. It's like more advanced. There are several steps between, but the basic is watershed transformer. No, it's not separated, the bone fusion, but the two vertebrae are separated, and here becomes one voxel wide space between them, which we consider as a bone fusion, this region. Oh, you mean this one? No. We just separated two vertebrae, and this is like the consequent Yeah, exactly. That's
Okay, thank you. Yes.
excuse me. Why is this surgery done? Yeah, maybe I can show it on the first slide. There are several reasons for this. Mainly, there are some degenerative diseases or tumoral diseases, and this surgery just prevents movement between two or more vertebrae in order to relieve pain or pressure of the usually intervertebral disc, which can be degenerated. So it's like, unfortunately, the occurrence of those surgeries is still increasing because of like sedative way of work of people, etc. Yeah.
Okay, great. Thank you very much for your presentation.
Thank you.
Thank you. So now I believe this is the end of tomorrow this morning session. And now we will have a lunch break and we'll be back at 1 o'clock. Yeah. And this afternoon we will carry on the presentation of Janet. Yeah. So I believe all of you have the lunch ticket.